Good morning, everyone. 3 years ago, I started a research project investigating methylmercury in the marine environment around our two offshore islands, the Rock and Great Duck, and this became my senior project. And this project really means a lot to me because it's my first research project, and I have wanted to be a field scientist studying whales and other marine organisms since I was a very small child. And I came to COA because of the Bar Harbor Whale Museum, which was operated by College of the Atlantic and Allied Whale. And at the end of the exhibits, there were examples of student work, scientific posters that had been presented at scientific conferences, and that has been something that I've been wanting to do for pretty much my whole life. So here finally was the opportunity of a place that I could do that, and here's the end result, which I have been able to present at various conferences. So pollutants are, in the marine environment and the terrestrial environment, are a global threat. While I'm studying mercury in the Gulf of Maine, it's not just coming from point sources from industry and things in our watershed, it's coming from global sources, so oceanographic input. You can see the local cycling in the Gulf of Maine here that's transporting contaminants and pollutants, but it's also coming from the Labrador Current, coming down from the Arctic, which was one of the biggest influences into the Gulf of Maine. It's also coming from weather systems moving across the country from west to east and bringing pollutants that are put out by emissions of industry in the Midwest. So it's a global problem. And methylmercury is just one of many toxic contaminants that are in our ecosystems, but it's particularly concerning because it becomes toxic in aquatic environments. It's deposited in a non-toxic form and then converted by bacteria to a toxic form. And that toxic form then bioaccumulates in the system. So organisms at the bottom of the food chain have lower levels of contaminants, and organisms at the top predators have the higher levels of contaminants. So it's a really concerning toxin in and of itself. Methylmercury has been previously studied in the Gulf of Maine in a wide range of organisms from blue mussels all the way up to seals and seabirds. It's mostly been studied in coastal environments just because there aren't many opportunities to study offshore pelagic systems, but we happen to have two of those right in our backyard, Great Duck and Mount Desert Rock. So these islands offer a rare opportunity to see what's going on in a pelagic environment and what contaminants might be there. So I spent an amazing summer out on Mount Desert Rock in 2012, there for the whole field season, and I studied methylmercury in seabirds and seals, which are on the island. So herring gulls and blackback gulls, this is a chick of them right here, they both nest on Great Duck and the Rock in the summertime. And seals, these are harbor seals down here, are found on Mount Desert Rock in the summer as well. And seabirds and seals are those type of organisms that are at the upper levels of the food chain, so we can expect them to have the highest levels of contaminants because they feed at a high trophic level of things that are more contaminated below them. So I took samples from these species. I took blood samples, egg samples, scat samples, and regurgitate. So this is me holding a vial of blood taken from a herring gull chick, and here's a regurgitate of what the adult coughs up to the chick. And so that is representing the fish, or what the levels of contaminants in the food, and then the blood, the egg, and the scat are what's coming from the animal itself. The levels of contaminants are dependent on what levels they're found in these animals' diets. So I had to know what the seals and the seabirds were eating, and if there were differences or similarities, maybe that would explain differences in contaminants in these animals. So I watched adult herring gulls feed their chicks from the lighthouse tower on both islands. This is me and Barbara out on the Rock. And I also washed seal scat samples that I collected to recover prey hard parts from them. So these bones here in this photo, these are the ear bones of fish. They're called otoliths. And these are two squid beaks here as well, and you can use those to identify the species that the animal is consuming. So here's a summary of what I found. On the horizontal axis here is the sample type. It goes from regurgitate, blood, egg, and scat. And on the vertical axis is the level of mercury I found in them, in parts per million. So the first result is that I found methylmercury in all of my sample types. And then the second is that there's a difference between all 4 of those sample types. So seal scat had the highest levels, and the gull regurgitate had the lowest levels. So then that is also reflecting the bioaccumulation through the system of the fish having the lowest levels and the other animals having the highest. There was only one obvious diet overlap that I found between the gulls and the seals, which was Atlantic herring. But then again, the natural history of these species is different, so seals are able to feed at deeper depths than the gulls are, so they can take advantage of different parts of the water column and prey that are there, so that could explain this difference that I found. I also didn't find a difference between years, so I sampled in 2012, and I continued in 2013 when I worked on our research vessel, the Osprey, collecting between the 2 islands. There wasn't a difference between 2012 and 2013. So what of this is that my results are pretty similar to studies that have been done all the way from 1972 up until present day. So it doesn't seem like there's been much change in the level of contaminants found in the Gulf of Maine and in these particular species. Again, there hasn't been consistent long-term monitoring in lots of places throughout the Gulf of Maine, but there's enough to kind of get a general idea that it doesn't seem like levels are increasing, but it also doesn't seem like they're decreasing. And again, that would reflect this is a persistent contaminant that stays in the environment for a long time. The other thing is that even far offshore, 25 miles out at Mount Desert Rock, you can still find contaminants. And that's a little bit depressing, but it's really important to know the extent to which we have affected the environment. And the methylmercury is not just a Maine problem. And this is really important to me. If it's not just coming from our watershed, we can't necessarily control what contaminants are found in the gulls and seals offshore. This is a global problem, and it's going to require collaborative and creative solutions. So I'd like to thank everyone that was involved with this study. I had a lot of help from many wonderful people. Thank you.